So hello everyone, welcome back to Hasfora Education. Today we will be solving some of the hardest MCQs of AS Biology. I have compiled these questions from 2024. So now let's begin. These are about 20 questions, so I'll be solving them slowly because they involve some unique concepts. So let's begin. The first question that we have is, which row describes the expected effect on Vmax and Km value when a competitive reversible inhibitor is added to an enzyme catalyzed reaction? So we know that when a competitive and reversible inhibitor binds, it binds to the it binds to the active site, and it means that the substrate can no longer bind to the enzyme. So if the substrate concentration is increased, then the Vmax can be attained and it can reach the Vmax. So there is no change on the Vmax when there is a competitive reversible inhibitor. When there is non-competitive irreversible inhibitor, there is effect on Vmax and the Vmax is reduced, while the effect of the substrate concentration at Km is that we know that the Km value increases and the affinity decrease. So they are asking substrate concentration at Km. So it means that the Km value will increase as the affinity for the substrate is decreased. So A will be the answer for this question. Let's move on to the second question. This is a bit tricky question because the common mistake in this question was that the students were actually confusing the Km value of the different lines with the other ones. So now let's have a look at it. The graph shows the effect of substrate concentration on the rate of reaction of three enzymes. So they have given us enzyme Y, enzyme X and enzyme Z. So what is the correct order of affinity of these enzymes for this substrate, starting with the enzyme with the highest affinity? So we know that for the affinity we are gonna need the Km value. Now first of all, let's take out the Km value of X, Y and Z. So the Km value is the substrate concentration at half the Vmax. So Vmax for the enzyme Y is this point. So half of this will be about the point. Let me just, so this will be the point of half Vmax, and just be very careful that when you are looking for the enzyme Y, you are also plotting on the line of it. So this will be the substrate concentration at of the enzyme Y at half Vmax. So it is about a 600. Now let's have a look at the enzyme X. So this is the Vmax for enzyme X and this will be the half of it. So let's check the substrate concentration. It will be about 150. So enzyme X will be 150. And let's check for enzyme Z. This is the Vmax for enzyme Z. Half of this will be about this point. So this will be about 200. Now you can arrange the Km value in the order with the highest Km value first. So it could be Y, then Z, then X, because this is 600 Km value, this has 200 and this is 150. Now you have to remember that affinity, the higher the Km value, the lower the affinity. So it means it will have the lowest affinity and it will have the highest affinity. Now can you arrange this in the order of affinity with the starting with the highest? So it will be X, then it will be Z, then it will be Y. So B matches our answer. So B will be the answer for this question. 12 says which row correctly identifies the weak and the strong bonds in the tertiary and the quaternary structure of a typical protein. So we know that in the tertiary or the quaternary structure we have four type of bonds. One is the covalent, and in the covalent we have the peptide, or you can also say disulfide. They both are covalent, okay? So covalent bond are the strongest. They are stronger than the ionic bonding, then which is it is stronger than covalent is stronger than ionic is stronger than hydrogen bonding, and hydrogen bonding is stronger than hydrophobic interactions. But out of these, only the covalent bonding is considered strong, while the other ones are weak. So for the disulfide, we will say it is strong, while the hydrophobic interactions and the ionic bonds, they are weak. So B will be the answer for this question. The fifth says which components of the bloods are present in the tissue fluid. So we know that tissue fluid is formed due to high pressure at arteriole end of capillary. And because of this, what happens is that the blood is squeezed out of the perforations of the capillary, and some of the components actually leak out. So only small proteins leak out and form the tissue fluid. The ions, or you can also say the mineral salts, toxic waste also, and there is a large quantity of water as well. So now we have the ions and the some proteins. So this and this will be correct. Okay. Now how are we going to decide whether the phagocytes are present in the tissue fluid or not? One thing you need to remember is that the tissue fluid passes through lymph, and lymph is the place where the lymphocytes and all the white blood cells are present. So when the tissue fluid passes through the lymph, what happens is that most of the phagocytes and the different type of white blood cells are become present in the tissue fluid. So that is why it is rich in WBCs, or the white blood cells. So phagocytes will be present in the tissue fluid. So A will be the answer for this question. 6 says the diagram shows the structure of 4 amino acid in aqueous solution. So which two structure have an overall charge? So now to look for the overall charge, what you have to do is that you have to look for the negative and the positive ions. So now you can look that it has a negative charge here and the plus charge here. So what happens that this positive and this negative cancels out each other. So in the end it has a neutral charge, so it does not have an overall charge. Now for alanine, you can see here we have the minus charge and we have plus charge here, so it will also have no overall charge. While the lysine, you can see we have the negative charge here, the plus charge here. These two will cancel each other, but there is no plus charge or no negative charge to cancel out the plus charge for this amide. So this will have a positive overall charge. Now can we eliminate the options which has alanine or glycine? Yes, we can. So alanine and glycine are eliminated. So we are left with the option C, but let's have a look at the last one, aspartate. So we have this minus sign here, we have this plus sign here. This will just you can say neutralize each other, but there is no plus charge to neutralize this negative ion, so there will be negative overall charge. So aspartate and lysine will have a overall charge, so C will be the option. Seventh is a tricky question because it involves a concept that is related to the lipids of triglyceride. So the table shows the number of carbon atoms and the number of carbon-carbon double bonds in the molecule of 3 triglyceride and the fatty acid they contain. The information is shown in the format number of carbon atoms and number of carbon-carbon double bonds. So they've actually given you the ratio. This is the carbon atoms number and its ratio to carbon double bonds, the total number of double bonds present in it. Okay, so which description matches fatty acid 2 of the 56 ratio 4 triglyceride identified in the table as X? So now we are going to be focusing on this one because what they have done is that you can see that for the first triglyceride, this is the first one, this is the second one, and this is the third one. For the first one, they have completed the fatty acid, their number of carbon double bonds, and their number of carbon atoms in each fatty acid. So they have completed for 1 and 3, but they have left for the triglyceride number 2. So now we know that a triglyceride is made up of 3 fatty acid chains and 1 trihydroxy alcohol. So this is the structure. This is how the structure of the triglyceride looks like. This is the part of the glycerol, and these are the 3 fatty acid chains. But one thing that you need to consider here is that they have not given you the glycerol, and we know that it is a trihydroxy alcohol, which means it has 3 carbon atoms. So now you can easily figure out the number of the carbon-carbon double bonds because they have given you that this triglyceride actually contains 4 number of carbon-carbon double bonds, and they have given you that the fatty acid 3 contains 2 double bonds. So how many double bonds are remaining? Only 2. So this will have 2 carbon double bonds. Okay, this is very obvious. But for the number of carbon atoms, what we will be doing is that we know that they have not given us the alcohol, so we will be subtracting 3 carbon from this, so it will become 53, and then we will be equating this 53 to 17 plus x plus 18. So then we will come up with x as 18 carbon atoms. The mistake that students will do here is that they will just equate 56 to 17 plus x plus 18, which is wrong because we also have to consider the number of carbon atoms of the glycerol. So we have to use our knowledge to it. So 18 carbon atoms, and it contains the double bond, so it will be unsaturated. So D will be the answer, and the rest of will be wrong. And you can also eliminate A and B straight away because you know that it contains 2 carbon-carbon double bonds, which is very obvious actually. Let's move on. Sixth says the graph shows various distances measurement taken from the start of the metaphase of mitosis. The graph are to scale when compared to one another. Now you can see that x is representing a distance time graph in which the distance is constant. This distance is constant, and then it is decreasing. So we have to look for a stage, or you can say an identity where the distance between two, it can be anything, the distance between the two is decreasing, and in the later stage, because you can see that it is decreasing after some time. After some time, it starts to decrease, while for the y it starts to increase. The distance starts to increase. Let's have a look. So which row correctly identifies the measurements for each graph? So now, so in the A we have the distance between poles of the spindle. So now you know that, for example, this is a cell. Okay, for example, this is a cell, and we have these centrioles at right angle at both of the opposite ends. So this is what the spindle actually looks like in the metaphase. So these are actually the poles. This and this is the pole, and they have given you that throughout the, you can say, the cell cycle. So the distance between these two poles remains constant. They do not increase nor they decrease. So these two will be incorrect. C says distance of centromeres from poles of the spindle. Now we know that in the metaphase the chromosomes align like this. Okay, they align like this. So what happens is, when the centromeres are there, when divided, they move to the opposite poles. So the distance between the centromere and the pole, it will decrease. Okay, so when we look at the x, it matches the graph. So these two can be correct. Let's have a look at the y. Y says distance of centromere from pole of the spindle. We have looked at it that it decreases, not did not increase. So this will be wrong. Now they are saying distance between two sister chromatids. So we know when the chromosome is actually divided like this, they move to the opposite poles, and when they move to the opposite poles, the distance between them should increase. So this will match the graph. So C seems to be our correct answer. And also I expect this question to be repeated this year, because it is the most repeated question I have ever seen in the past years, as well as in the 2024. So I expect this question to come again. The other question says the electron micrograph shows a type of blood cell. So now they have given you the hint that it is a type of blood cell. Now it can be a red blood cell, or you can say it can be a white blood cell, but we have to identify from its structure. Let's have a look. So now first of all, before having the options, you can see here that there is abundant amount, almost all of the cell is covered in rough endoplasmic reticulum. You can clearly see it. Okay, so abundant amount of rough endoplasmic reticulum. So what it means? It means that it is highly involved in protein synthesis. While you can also see that the nucleus, which is like a lobe structure, like that of the phagocytes. Now they are saying what can be concluded from from the electron micrograph. The first one says the cells secrete products that are toxic to pathogens. No, this is incorrect. The cell synthesizes a large quantity of proteins. Yes, this is what we actually said. So B can be answered. Let's have a look at the C and D. The cell synthesizes large quantity of antigens. Now this can't be correct. Why? Because we know that it is a type of blood cell, and it will actually secrete a large amount of antibodies. So the cell digests pathogenic bacteria. This cannot be actually very obvious. In case if it contained lysosomes that were visible in the diagram, then we could actually say that it digests the pathogenic bacteria, and then it digests it through the forming the phagolysosome, or the you can say the phagocytic vesicle. So this will also be incorrect. So B will be our answer. So 13 is a very tricky question because you have to use your knowledge of the plants to actually answer this question. So the diagram shows how nicotine is transported from the blood plasma in the cell using a type of cotransporter mechanism. So do you remember that where did you study the cotransporter mechanism? We studied this when the sucrose moves from the source cell to the companion cell. So between the source and companion cell. So you can see that the protons move down their concentration gradient out of the cell. This is moving out of the cell, but nicotine is transported against its concentration gradient into the cell. So in the phloem tissue, they have already provided you the main idea. So there is a co-transporter mechanism that moves sucrose into cytoplasm of the companion cell. So which statement correctly describes a similarity between the co-transport of nicotine and the co-transport of sucrose. So what actually happens is that, for example, if the protons are moving down their concentration gradient, it means that they will be in high amount of H plus ions here, and there will be low amount of H plus ions here. So now what actually happens is, for example, the nicotine is transported against its concentration gradient. So now what will happen is that, for example, the H plus ion will first move against their concentration gradient, and then when they will become abundant there, what will happen is that when they will become abundant here, so what will happen is that, sorry, I have written it opposite. This will be, let me just correct it. Okay, so this will be in high amount here and low, because we know that it moves from higher to lower concentration gradient. So for example, the H plus ion will first move here, they will first move here, and when they build up, in abundant amount, then they will move down the concentration gradient out of the cell. Okay, so this is the same mechanism how we actively pump the H plus ions into the source cell, and then when the hydrogen ions build up in the source cell, then it co-transports with the sucrose. So the B is the correct statement, which says the protons are transported through the co-transporter proteins by facilitated diffusion, because we know that they are polar because they contain the plus ion charge, and also they are passive, so they will use facilitated diffusion. While you can have a look at the other ones, the cotransport proteins generate a proton gradient by moving protons out of the cell by active transport. We know that it is moving down the concentration gradient, so it will not be active transport. C says that protons move through the cotransporter proteins in the opposite direction to the movement of nicotine and sucrose. So this is incorrect because, for example, it is when it is co-transported, it will move together with it. So D says that the co-transported protein uses energy from ATP to transport protons. No, because we know that it uses facilitated diffusion to carry out the transport using down the concentration gradient. So it will not require energy or ATP. So B will be the correct answer. So which aspect of the enzyme activity can be compared by the Michaelis-Menten constant? So we know that Michaelis-Menten constant is the Km value, and Km value is the substrate concentration at half the Vmax. So, and it is actually used as a measure to deduce affinity. So A says activation energy of a reaction with or without an enzyme. This is incorrect. Affinity of different enzyme for their substrates. This can be correct. Affinity of enzyme at different substrate concentration. This is incorrect. And also They have told the maximum rate of reaction Vmax at different temperature. This is incorrect because the affinity don't depend on factors like pH, temperature, and substrate. However, we can say that it can be used to reduce the affinity of different enzymes for their substrates. So B can be the answer. So 23 says which statement correctly describes the process of translation. So we know in translation what happens that the mRNA binds to the ribosomes and then the tRNA having the amino acids they bind to uh, the the anticodon binds with the codon via hydrogen bonds and the amino acid at the 3' end they just grow a polypeptide chains and they are linked up using peptide bonds. So let's have a look at the statements. The nuclear sequence on the mRNA's molecule is used to produce a specific amino acid change. This is correct because the anticodon is specific to the codon and it will actually determine which amino acid will attach. So this will be correct. Second says a section of DNA is copied into mRNA molecule by RNA polymerase. This is incorrect because it is talking about transcription where a template strand is used to produce a complementary mRNA molecule. So this will be incorrect. 3 says a polypeptide is produced because anticodons on tRNA molecule attached to mRNA codons through peptide bond. This is incorrect. They are bonded through hydrogen bonds. So 1 will only be the option. So C is the correct answer. So fourth says which type of cell will have the highest proportion of its volume taken up within the cell structures bound by a single membrane. So we know that all of these are actually single membrane cells. And now one thing that you will have to think here is that what is the most abundant structure that you have seen in a cell. So my answer will be secretory vesicles because in the animal cells they are present in an abundant amount. And now why are they called secretory vesicles? Because they secrete either the enzymes or you can also say or mucus, just like in the trachea or the bronchus. So now to secrete the mucus, what is actually needed? The goblet cells, they produce the mucin, and the mucosa glands which produce the serous fluid to form the mucus. So we will actually need the goblet cell in more amount to actually have to actually we can say release more mucus. So it will actually take the highest proportion or the volume of the cell structure in the form of secretory vesicles because in the secretory vesicles the goblet cells are the main component that secrete the mucus. So the other options are actually wrong because you can say that the red blood cells do not take up much volume. They are very small in the size and also how are they going to be taking up much volume? While for the goblet cell, they are going to take up the volume itself and also for the mucus they release. So B will be the correct answer for this. So this was actually a very tricky question. This was also reported in the examiner report as being mistaken or misled by the question by the students. So many of them actually circled the A or C. So what causes the phosphate heads of the phospholipids to become polar? So let's have a look at the options. The phosphate heads are joined to water molecule by the hydrogen bonds, but this is correct. Okay, this is a correct statement. But if you look at the question, they say that what causes the phosphate heads of the phospholipids to become polar? Hydrogen bonding do not actually cause an you can say a molecule to become polar. It will increase its strength, and it will also increase, you can say, the heat required to break its bonds, but it does not cause any of the molecule to become polar. So this will be incorrect statement. The phosphate heads are insoluble in water. This is wrong because we know that it is hydrophilic and it is soluble in water. The phosphate has become ionized in water. Yes, this is correct because we know that this is the structure of the phosphate. This is the structure of phosphate. So what happens is, for example, this has a change in electronegativity, and this has partial positive and partial negative charges. So what happens is that when they dissolve in the water, they ionize. For example, one of the bonds here can break and it can form a negative charge. So this is the reason, and it can actually attach with the this is the structure of the water. Okay, so this can actually make the bonds here like this. So this is how actually they link up with the water in the water. The phosphate has actually link up with the water, and it can actually break to produce ions. So when they produce ions, they become polar because there is a change in the polar. Actually means change in the polarity, change in polarity, or you can also say the there is a partial negative or partial positive charges on the species. So C will be the correct answer. D says the phosphate has a joined to water molecules by covalent bond. This is incorrect because they are hydrogen bonds, not covalent bonds. So hydrogen bonds are a category of intermolecular forces. So C will be the correct answer. The next question says which statements are correct reason for how animals cool down in hot environments using latent heat of vaporization of water. Some animals lie down and roll in wet soil. So now what will this measure do? This will make their skin wet, and then the wet skin, the water molecules on that skin, what will happen that they take up much heat to evaporate. And you also know that evaporated also has a cooling effect. So when they take up much heat, so more of the heat is absorbed and removed from the body in this in the eh, you can say in the shape of uh, less number of volume of water. So this will be a correct statement. So fish move into deeper water. This is incorrect because if they are already in water, so what is the point of moving into deeper water? Because the temperature will remain the same. Okay, so this is incorrect. 3 says some animals lick their fur to make it wet. So for example, if they make their fur wet, what happens? It will be the same effect as if they roll in wet soil. So if they lick their fur to make it wet, so the molecules of water take high amount of heat to evaporate. So this will also cause the same effect, and it will actually take up much less volume will be used to remove high amount of heat energy. So some animals breathe quickly with a wet tongue hanging out of their mouth. This is the same effect as this, because in this case their tongue will be wet and the water molecules will be evaporated from their tongue. So 4, 3 and 1 are correct. So 1, 3, 4, C is the correct answer. Next question says two different enzymes P and Q were investigated to find the optimum pH for the each enzyme. The result shown, the P works only in acidic conditions. So this is acidic. P is acidic, and Q has an optimum pH which is slightly alkaline, and this is alkaline. So which graph shows the correct results? Now in the first graph you can see that they have shown the pH of the P as neutral. So this will be incorrect, and they have shown the Q as acidic. So this is completely opposite to what they said. Let's have a look at the other weird looking graphs. So C is actually, you can say, they have plotted the pH on the y-axis, and you can see that the P is again on the neutral pH. So this will also be incorrect, and Q is again to the against to the acidic side, so this will be incorrect. In the D, what they have done is that the maximum pH is 7 and all of them is acidic, so this will be completely wrong. Now one thing that students will confuse in the B part is substrate concentration at 1 minute. So why have they drawn this inverted graph of A? So what actually happens is that when we measure the rate of reaction, we either measure it through the increase in the product concentration or the decrease in the substrate concentration, because the substrate will form into the product, so it will decrease and over time. So if the rate of reaction is increasing, it means the substrate will decrease. Okay, so I hope you get the concept. So now let's have a look at the pH. You can look here that it is acidic, it is below 7, and for the Q it is slightly alkaline. So B will be the correct option. So second last question says the diagram shows an outline of the mitotic cell cycle. So they have numbered the stages. So now you can have a look that the cytokinesis, if the cytokinesis is taking place here, so what will actually be the clue here? So if it is placing here, so now it means that mitosis will be happening here, because we know cytokinesis is the end of the mitotic cell cycle. And if this is the mitosis, then this will be the G2 phase, this will be the S phase, and the 3 will be G1 phase. Now which number stages of the cell cycle include a period of time when each chromosome consists of sister chromatid joined by a centromere? So sister chromatid joined by a centromere, the clue they want to give you here is that at what stage will it have a look like this. So we know that this only happens in the mitosis, in the prophase, when it condenses to produce sister chromatids, and also after the S phase, and also after the S phase, because what happens is before the S phase only this is present. Okay, only one chromatid is present. In this S phase, the DNA replication occurs. Although this converts into this, but it is actually one chromosome. Okay, you should have this idea. And prophase, the prophase, and in all of the mitotic cell cycle, this is the cell shape. Okay, until unless when it comes to the anaphase and the telophase, but generally it is the structure of the chromosome in the mitotic cell cycle. So we will have our option as 1 and 2, because after the phase and in the mitotic cell cycle. So A will be the answer for this question. Now the last question that we have is, a student drew a sketch to show the formation of tissue fluid. Which label is correct? Okay, now so we have to have a look on these labels. So blood enters capillary and the pressure increases. Now this is the point that invalidates this option. Why? Because we know that the pressure is absolutely high at arteriole end, or you can say when it is coming to the capillary. So pressure is indeed high at the arteriole end, but what they have said is that the pressure increases, it do not increase. It is already a high pressure. So you have to be just very carefully looking at these points. So B says red blood cells are water move into tissue. This is correct. Water moves into the tissue, but red blood cell do not moves into the tissue, because the perforations are about the same size of the red blood cells, so they cannot squeeze through it. So B will also be wrong. C says higher water potential in tissue fluid, so water moves back into the capillary. This is correct, because I have told you a high amount of water moves into the tissue fluid, and at the venous end, This is how the down the potential water potential gradient it moves again into the capillary. So C will be a correct option. Let's have a look at the D. Higher volume of the blood returns to vein. This is incorrect because when the tissue fluid forms and some of the volume is leaked into the tissue fluid to form the tissue fluid, so 85% of that is returned at venous end, which is a little bit lower, and the 15%, the remaining 15% is returned later via the lymphatic system, because this tissue fluid then goes from the lymphatic capillaries to lymphatic vessels, and then they drain into the main bloodstream. So there will be lower volume of the blood return at the venous end. So this will also be incorrect. So C will be the correct option. So we have completed the paper. I'll also be attaching this paper in the description down below. If you have any specific queries, you can let me know down in the comments down below, and I'll try to answer that in the comment section. So I'll meet you in another video of the final tips before the paper and the probably some of the expected questions that I think it can appear this time in the paper. So I'll be meeting you in that video. Till then, goodbye.